This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 573, recorded on November 8th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it's blustery here. It's cold, and actually, let me get the current temperature because I know everybody's dying to know. It's 35 Fahrenheit, 2 Celsius, but it's blowing 15 to 20 knots out of the north. Wow. And unpleasant.
Here's 4 degrees Celsius, mostly sunny. It's a nice day, but all of a sudden, overnight, it got cold.
Yeah.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. I'm always surprised that you guys don't have really similar weather, but I guess there is some distance and maybe even some hills separating you.
Yep, there's some land separating me from the water, and Vincent's right on the water, so a bit of a difference.
Yep. But
we've got 48 degrees, and that's 9 Celsius, and overcast and kind of off and on rain/drizzle. Good day to be indoors. I spoke to Kathy about 45 minutes ago. She's going through the ASV. I wouldn't call them archives. It's the files from the last, 20 years or so, with Lorena Passarelli and Andrea, her assistant, trying to figure out what to keep and what to throw out, which is why she can't be with us. She wishes us all well and says they got a dusting of snow last night.
I can't believe that, going through junk trumps TWiV.
Well, what can I say? Actually, Lorena, it's because Lorena was there.
Yeah, she went there. I got it. She came from
Lorena was there for a seminar. They took advantage of this. I mean, this is one of those things, one of those really painful tasks that's got to be done.
Got it. All right, Dickson will be here later, but we do have an in-studio guest here in New York City. He is from Yale University. William Summers, welcome to TWiV.
Well, thank you very much. Happy to be here.
Can we call you Bill? Does that work?
Bill, that's fine.
Okay. I saw you talk at ASV this past summer. A wonderful talk about the history, or part of the history of virology, and I thought, let's get you here, and maybe some of our listeners can get to learn some of what you said. So here you are. And if you like what we do here on TWiV and all the other podcasts, consider supporting us financially. Go to microbe.tv/contribute. Let's start with your personal history, Bill. You just retired 2 years ago, right?
That's right.
So go back to the beginning. Where are you from?
Well, in the beginning, I come from a small town in southern Wisconsin, actually the famous site of Paul Ryan, whose grandfather actually hired my dad as a lawyer. But I was planning to grow up and be a small-town doctor, and went to the University of Wisconsin, where things changed. I majored in math only because it was easy, but was firmly planning to go to medical school, which I did. And
at Wisconsin also?
At Wisconsin, yeah. I mean, I guess the idea was that if I was going to practice in a little town in Wisconsin, why not get to know the local docs through that contact? So one summer, however, the NSF offered $600 summer fellowships to work in a lab. And I decided, well, I wanted to hang around Madison because I had a girlfriend around. And, these are contingent things, as the historians say. And so I got into a lab, and by the end of the summer, I was hooked on lab work, and medical school started to recede in my interests. The lab was in the radiation biology section, and I was doing work on mouse tumors. And
were you still in medical school at the time?
I was still in medical school. They had a program sort of a forerunner to the MD, PhD, where you could get a master's degree by splitting your second year of medical school. Mm. And so I signed up for that. And fortunately, the Wisconsin Alumni Research Foundation, WARF, supplied me with a stipend for the year, which was great. And I got my master's in radiation biology. Along the way, I developed a raging allergy to rodents, and
get some warfarin for that.
Yeah, right. And as the immunologist, the allergy specialist said, we could try to desensitize you, but for a whole career, you ought to look for something else. And I think that's right. I mean, I could be living on antihistamines or something, but and at that time, a man working on radiation genetics, Václav Szabalski, had just come from Rutgers to Wisconsin, and I went and talked to him about working for him in radiation biology, and he took me on as a student, which was interesting because at that time all the rest of the students in the department had been assigned to advisors. Whoever, quote, needed a grad student would get their pick of the new class. And I remember the first week I was there, I was assigned to do a joint seminar with Gunther Blobel, who was a grad student, and a few other people. And their universal reaction to me was sympathy. That is, that I had gotten assigned to Václav. And when I said, "Oh no, I chose him," they just didn't understand me at all at that point. But it turned out it was a really fruitful interaction. And I tried a few experiments. He suggested that there was this new phenomenon called restriction and modification that he and Ray Erickson, who's now a professor at Harvard, were working on. And he said, well, why don't you figure out what the mechanism of restriction is? And he suggested I do a Hershey-Chase experiment to see how far the genome got. Mm. And it turns out I couldn't grow phage worth a darn. And after 6 months, I gave up. Missed a Nobel Prize probably, but many of us can say that, right? So I ended up doing my dissertation on the radiation biology of DNA, using B subtilis as a system where you could, on one hand, look at phage DNA, and on the other hand, look at transforming DNA from the bacteria. It turns out one is sensitive, is inactivated by double-strand breaks, and the other is inactivated by single-strand breaks. So it suggests that the mechanisms are a little different. And as a phage, I used a very small one that I could find called Phi 29, which was subsequently very famous. And those phages were isolated by John Spizisen and Bernie Riley at the University of Minnesota.
Spizisen broth.
Yes, that's right.
I remember that sitting on the shelf.
Interestingly, I spent a couple of weeks in Spizisen's lab where Bernie taught me how to finally grow phage properly. Uh-huh. And then he came to our lab where I actually did the physical studies on their collection of subtilis phages. But
wow.
So it was actually interesting when I was at the ASV meeting, the micro department at Minnesota was having their 100th anniversary, and I piggybacked on that one and met a number of people I hadn't seen for, again, quite a long time. And
the names you've just dropped are like amazing so far.
Interestingly, Spizisen was an undergraduate who worked in Max Delbruck's lab at Caltech. So he was not a favorite of Max's because he quit and went to medical school. Mm-hmm. And but that was a whole nother story. So I was planning to go, after finishing medical school, go to a pathology residency, and because I was liable for the draft, and the way to get out of the draft was to take a residency and then go into the military. And I couldn't find a pathology residency that suited me, and so I decided, well, I'd take my chances and go be a postdoc at MIT, where I spent. I was going to spend a year with, or 2 years with Cy Leventhal I thought I'd learned something about proteins. Well, no sooner had I arrived than Cy said, well, we're all moving to Columbia.
Right.
And he got trapped in the biology office for a month while the students, had to sit in, and we hardly ever saw him.
This is 68, right?
This is 68.
Yeah.
And
Mark Rudd, right?
yes, right.
Oh, my gosh.
I mean, this is all by way of saying, you don't plan careers.
No.
Well, I agree with that.
And so within 2 weeks of arriving in MIT, and Boston was scary enough, but New York was really scary for a Midwestern boy. And I started looking for a job. And Cy said, well, why don't you count how many genes make antibodies? Which was an interesting problem at the time, right? He had heard that I could do DNA hybridizations, and he had also heard that somebody had found out, that there's these myeloma proteins, and they make just one major protein. And because hemoglobin in reticulocytes, the message has been just isolated, why don't we do that with myeloma cells? We could just take this message and hybridize it to saturation and count how many sites there were. On paper, it sounded fine, but I ended up killing an awful lot of mice with tumors with P32 to label up their RNA hot enough. And again, this is an experiment that I couldn't make work. And another Nobel Prize missed.
Right.
So you're labeling mice with P32.
Right. I mean, I just watched them waste away as I shot them up with 10 millicuries every day because I was trying to label the message to the same to the constant specific activity so that I could use the global RNA specific activity to be the equivalent of the message. And
wow.
So,
between that and the rodent allergy, this is not looking good.
No, well,
for the uninformed listeners out there, P32 is a radioactive form of phosphorus that is commonly used as a radio label for nucleic acids, and it's got a kick.
Yeah, that's right. That's right. Well, I could just watch these animals sort of waste away from radiation poisoning, but then the coup de grace came when MIT was not known for its animal care facility. And there was a neurobiologist named Jerry Letvin who had a lot of cats that he was doing neurobiology on. And that was in the next room. And I came back after Christmas break, and the lab animal technician had a note on my mouse cages saying, "Eaten by cats."
Oh,
Apparently somebody had left the door open, and the cages somehow they escaped. And at that point, I decided this was an ill-fated experiment.
So, did any of the cats eat hot mice?
They probably were also labeled by then.
Yes.
Right. I did do an experiment which actually was interesting and got some. We published it in Nature, where because I had worked on phage with Jabalski. Dave Botstein was just down the hall, and he and a few others had just put together this notion of the rolling circle model for replication of viruses, of phages, the Phi-X174 in particular, single-stranded phages. And one prediction of that model is that only one of the two strands ends up in the virus. One is just a template and thrown away. And we wondered whether indeed that happened with double-stranded phages. Because actually Jim Watson had published a paper long before this about how, because at that time only 50% of the label of a parental phage made it into the progeny phages. We now know probably just for technical reasons. But because the 50% notion was found by several labs, people thought maybe it had some meaning. And Watson said, well, maybe one of the two strands is template and never, used in the progeny. And so we could separate the strands of the DNA, and I found that no, the progeny had equal amounts of parental label in both strands, and that was an old problem that nobody was interested in, but we solved it. And so by that time, I had gotten a job at Yale, and that really came about because, as I said, I started looking for a job as soon as I realized I would have to go to New York. And, well, I interviewed here, and Cy had this beautiful 8-room apartment overlooking the river and so on. And he was perfectly happy to be here. And then he told me that they'd make me an assistant professor to run the pre-med program between Columbia Medical School and the biology department. And I had an appointment. Paul Marks, who was the dean of the medical school, arranged an appointment for me in the medical school. And they said, well, the salary's $8,000. Yale had offered me $14,000, and several others up to $18,000. So, Columbia was a little bit behind. And when I told Cy that, he was appalled. He said, well, we're going to have to fix the salary scale.
Never change.
But
so what year was this?
This was actually the fall of 1968. And I had the summer before I had met Frank Hutchinson, who was the chair of biophysics at Yale, on a trip on the steamer on the Rhine down the river. We went on a tour after a meeting, and I happened to run into him, and we spent a day drinking wine together on the tour boat. And he was one of the few people I knew who was a shaker and mover, who might know about jobs. So I wrote and says, do you know of any jobs that are available? And he said, well, come down and Yale and talk to me. Well, I went down to Yale and went home with a job. And it's not done that way these days. There was no advertising. There was nothing. And today
there are also no jobs. So that's all right.
Right, yeah.
It's all by committee today.
Right. Well, this Paul Howard Flanders had just gotten a very large center grant from the NIH to study basic cancer research having to do with mutations and that sort of thing. And they thought I was going to continue my antibody gene counting work, but I had already sort of given that up as undoable given the technology. And I went back to viruses and basically spent the rest of my career doing viruses, first phage and then herpes viruses.
So how did T7 happen?
Well, T7 turns out to be the colophage that people were first noticed you could isolate easily the intact DNA. And so I was using that for some of the radiation biology work after 529, which is about half the size of T7. But because Chabowski had found that you could separate the DNA strands by a technique of cesium chloride density gradients in the presence of polyguanylate acid. And the thought was these poly-G stretches somehow would bind to sites on the DNA and make it more dense. And it seemed to correlate with transcription. So that in lambda, the strand that had more poly-G sites had more transcription by hybridization. It turns out T7, when we separated the strands, one strand has no poly-G binding sites on it. So the prediction would be that that strand wouldn't be transcribed. And so I did that experiment after I'd finished my thesis for a couple of months. And it probably was the most satisfying experiment of my life. I separated the two strands quite cleanly and then did the hybridizations of labeled message. And I put it on the counter, and there was like 40,000 counts on one strand and 4 counts on the other. It was the most dramatic difference. I mean, you didn't have to fuss with background. It was background. It was almost publishable instantly.
So is this one of these true, unrelated phenomena? Because there's all this stuff about poly-G and everything else, and everything it turns out to be right, but not for that reason.
Not for that reason, right? Exactly. We still don't understand why the poly-G, what the poly-G binding sites are all about. I mean, I spent a couple of papers trying to do the thermodynamics to get some sense of whether it was regular base pairing or what, but and now they have the sequence. You look and you're not even sure still where these sites are, but we could do, we count them and so on. But
so did you spend most of your phage time on T7, or did you do
others? Yes. Yeah. And then I got interested because again, wrong, right, wrong sort of thing. In T4 phage, there were this problem of why late transcription doesn't happen if you block DNA replication. And it was thought, well, maybe there's something about the template, or that have to have freshly replicating DNA in order to transcribe it and so on. And because I had worked on T7, I said, well, I knew that Rudi Hausmann in Germany had just isolated a number of mutants that were blocked in DNA synthesis, and the so-called DNA zero mutants. And I got some of these mutants from him, and because I already had the RNA, and it turns out that these DNA zero mutants did not make late transcription, just like in T4. Turns out for a totally different reason, however. And so we wondered what was going on here in these phages. And at that time, I met Dick Burgess, who was in Watson's lab, and his wife and my wife had been in the same lab as undergraduates, his wife was an undergraduate, my wife was a grad student in Wisconsin, and that was the connection. But Dick was working on RNA polymerase, and so he said, well, why don't you look at RNA polymerases and see if that's modified? And because he had just discovered sigma factor, and the thought was, well, maybe T7 makes a new sigma factor that changes the transcriptional program. And I think this is really interesting because phages are like mini embryological experiments. They have a genetic program of development. And I think that we were all thinking, well, this is a model for development. How do genes get made in the right sequence? And so we looked, and one of the criteria for study that Burgess had come up with RNA polymerase was the fact that there were certain mutants that were resistant to the drug rifampicin, and I was adding rifampicin then at different times to see
rifampicin being an RNA polymerase, coli RNA polymerase inhibitor.
Yes, thanks. But I found that within 3 or 4 minutes of infection, all the transcription that went on was resistant to rifamycin. And that suggested that whatever was going on, we had a way of purifying in the late part what the RNA polymerase transcription was all about. And it turns out it was not E. coli polymerase anymore. It was a new molecule. And Bill Studier had just basically saturated the T7 map with mutants, so we could basically screen the whole genome and find out. And sure enough, there was an early transcription product that was needed for late transcription, and it turns out it was an RNA polymerase. And at that point, it became interesting to a lot of people. And so I've always felt that if a problem is already well covered by other people, you might as well just go fishing because the problem will get solved. And the idea of being first is not exciting to me. The idea of being in a field where nobody else is working and finding it out is interesting. And I think you can justify it's more cost effective in terms of your time. So we sort of transitioned into working on herpes viruses because there was very little molecular biology of herpes viruses in the early 70s. And herpes sort of looked like T4. It was easy to grow. We thought it was not dangerous. And so we gradually morphed into that. And there were a lot of interesting problems. But again, all of a sudden, we were sort of swamped from behind by 500 new labs working on it. And when we started, there were 3 or 4 who were taking a molecular biology approach. So that's sort of a long-winded.
And then a number of years ago, you got interested in history of.
Well, that comes from teaching. Those of us who teach realize, you teach what has happened and how we got here. You don't teach where we're going. And so how we know what we know became sort of interesting. And I started to co-teach with a historian of science when I was teaching medical school biochemistry. And I found what he was teaching was a lot more interesting than a lot of the sort of standard textbook stuff. And the students can read the textbook. Biochemistry is one of the fields where there's really great textbooks for the facts. But then I found that scientists tell campfire stories rather than history. And I started hanging out with the historians, and I realized they have a technique, and they have techniques, and they have standards, and they have methods for verifying things and for extrapolating over the gaps when you don't have things. they're not just stories that your teacher told you. It's grounded in data. And I found that scientists in general would never publish a paper about science without data, but they certainly will publish a lot of historical stuff without data. And so I sort of decided I was going to, I don't know, not make a revolution, but try to do a little bit better. And so little by little, I got into this. And I actually, again, serendipity plays a big role in these things. A friend of mine in Florida, an old TWiV person, Donna Duckworth, had published a paper on who discovered bacteriophage. There was sort of a conflict, a controversy about this. And she mentioned that at one time, one of the two candidates for discovery was a guy working at Yale named Félix Thérel, French-Canadian. And that she had tried to get into the records at Yale, but the dean said they were closed. Well, I was waiting for the dean, and he was running half an hour or an hour late. And I was sitting in his office, and I asked his secretary one day if she had faculty records from the old days. And she said, "Oh, yeah, they're all over there in the cabinet." And so, well, I knew her, and she said, "What do you want to see?" So I pulled out this file, and it turns out it was quite interesting. And I realized I now had some data about this guy's time at Yale. And he had spent 5 years at Yale. He basically got fired because he had a company. Now you get fired if you don't have a company, but he was spending half time in Paris, basically. And finally he agreed to even take a half-time salary. But eventually, he ended up leaving and actually going to Tbilisi in Georgia, where he established the phage center there, which to this day is now the center of phage therapy.
Yeah, that's right.
So,
that's right. But he did discover the phage not at Yale, somewhere else.
No, he discovered that actually when he was working as an unpaid assistant or volunteer actually at the Pasteur Institute in 1917. He had a very long and interesting story, which
when was he at Yale?
1928 to 1934.
Okay.
And we talked about Twort and Durrell on a TWiV some time ago. I don't remember what episode.
Ancient history.
Very old.
Yeah.
Yeah.
Well, I think we talked about that in like 1920.
When it was news.
Yes.
And so Twort was the other guy, right?
Yes. Twort was an interesting guy. He was an MD working in a veterinary micro department in London. And he was trying to grow vaccinia in cell-free culture. And vaccinia samples being taken from pustules of skin are contaminated usually with micrococci and so on. And he noticed that the micrococci contaminants often would be lysing off. And the vaccinia has nothing to do with it. It just was why he was doing the experiment. Yeah. But he called this glassy transformation. Yeah. And because they look sort of mucoid and he couldn't grow any bacteria out. They were dead. And so he published one paper where he speculated what might be the cause of this. And that was it. I mean, if you read the paper, it's sort of speculative and not clear, whereas Durrell's 2-page paper, it nails it. I mean, he describes the plaque assay the way we do it now. He interpreted it as these, microbes that infect bacteria and sort of microbes of microbes and so on. So,
Twort also at the end says, "I can't continue this work because of lack of funds."
Right.
But I hope somebody else does.
Right. Well, he also being an MD got drafted to go in the World War I. And so he left for a couple of years and didn't have opportunity.
So I found it here, TWiV 145. August 16th, 2011. It was titled The Invincible TWiV because Vince wasn't there.
That's right. That was
right.
Alan and I did this ourselves, and we discussed the
that's why I remembered it so well.
Discovery of bacteriophage.
Okay. Yeah.
One of the things that's interesting, and I just wrote a piece about this, was what it means to discover something. And I think there's an overlooked paper by Thomas Kuhn, which was the prelude to his book on the structure of scientific revolutions, where he talks about what discovery means. And it's not an event, it's a process. I mean, he says, for example, when you say who discovered oxygen, it's not the guy who had a bottle and put a cork in it for the first time. You got to put it into context so scientists understand what it is and put it into a framework of understanding and meaning. And so, it starts with some sort of observation of weirdness, and it ends when everybody's on board with what the case is. Mm-hmm. And you can't go and say, well, today it went from being undiscovered to being discovered.
Right.
And I think that's the case of phage.
So was I forget the sequence. Because Twort and Virol were more or less contemporaries. Is that correct?
Twort published his paper in 1915 and in Lancet, and Virol published his paper in 1917 in Compte Rendu.
So was Virol aware of Twort's work?
No, quite clearly not. And what happened was that, again, things that are non-scientific in a way. Twort basically took on Jules Bordet, who had just gotten the Nobel Prize for immunology, and said that really the reason people recover from disease is not because antibodies are produced, but because their phages colonize their body and kill off the organisms. And this was supported by the fact that if you do serial titers of people, say, with dysentery, that as they get better from the dysentery, the phage titers go up. So there's more phage supposedly killing off things. And that led to the whole idea of phage therapy. And Virol, unfortunately, published a book in which he had a section called The History of an Error, in which he talked about Bordet's work. And this infuriated Bordet's group. And they looked for anything to beat up on Virol. And they found this paper, and they didn't attack the science of phages, they attacked his priority. Right. And this argument about priority went on for about 10 years. And it actually ended up in the Parisian courts.
Wow. So there is an aspect of this I wanted to touch on. So by the time these two have found phages, we have had viruses since the late 1800s, right?
Right.
And you have a slide in your deck from ASV called The Technological Origin of Virology. We talk about filters. Talk a little bit about filters.
Right. So viruses, as I think many people know, started out as being anything that it was a poison that's transmissible and so on. And it had to do with the change from, evil eye and godly influences to material aspects of infection, stuff.
This is way before discovery of what we now call viruses.
Right. But they would take pus from an infected animal and put it into another animal, and the animal would get sick. So it was the stuff of the pus. And they began to analyze what was in the stuff. And
it wasn't demons.
And it wasn't demons anymore. It was at one time, but now it became stuff. And so little by little, in the middle of the 19th century, they discovered that you could see these things under the microscope called bacteria and protozoans and so on. But along towards the end of the 19th century, they felt that, they needed a source of sterile water. And one of Pasteur's assistants found that if you filtered water through a terracotta, like a flower pot, the water goes through, but the bacteria that cause the disease are held back. But they found that some things went through, but you couldn't see anything under the microscope. And so these things that went through were a kind of virus. It was a subset of viruses, because the viruses that caused tuberculosis and anthrax and so on that were bacteria, those are also called viruses. In
fact, Pasteur said everything's a virus.
All microbes are viruses, or all viruses are microbes.
That's right. All viruses are microbes.
Right.
Because he had established that bacteria, or you can see them, they cause disease, they ferment and so forth. Right.
And they, make cheese and wine and cause diseases of.
Right. This is important because today's view of virus is very different.
Right. Well, so it had to do with the disease and biological things. The end point was disease, right? Or lesions on a tobacco leaf or something. Mm-hmm. And the fact that they could be passed through a filter said something about their size. And so we got a classification of filter passing and filter retaining microbes, if you will, or viruses. And so they called the one kind of disease was filterable viruses. That is, they pass the filter. And diseases like the tobacco mosaic disease, which you could easily take and transmit by grinding up a tobacco leaf and putting it on the other tobacco leaf. That was a classic, inoculation experiment.
Yeah.
And foot and mouth disease, transmittable in the animals. So whether it passed the filter or not was one of the first tests. So this really said it was a technology of the filter, which allowed you to have this conception of filterable viruses. It wasn't any new belief in the fundamental aspects of what was going on. It was a technological decision. Right. Just like microbiology is a technological thing. It says it's small. Right?
Yeah. But at some point, someone realized that these filterable things, viruses, you couldn't grow them in a broth, right?
Well, that was one of the things, is that they tried to grow these things away from the animal. And that was one of the key things that Virol noted, was he said these phages, he thought they were basically representative of viruses, were obligate intracellular parasites. You couldn't grow them outside the cell. And I think one of the things, if you look at viruses, they're sort of peculiar in that the definitions are, especially in the early days, are what things aren't. They're not culturable. They're not visible. And that's philosophically sort of unsatisfying.
Yeah, sure.
And also you miss things. I mean, if you have a virus that doesn't have any effect, the way you're assaying it, I think you pointed out before, assays are really crucial. Mm. And now we of course know that there's all these unculturable organisms, probably many more than we actually know how to culture. But until recently, we had the hubris to think, we know how to culture most of them. And
it's interesting, though, that I'd never thought about this, that concept of characterizing stuff by what they aren't. But because if it's a total novel thing, you don't have anything to compare it to. The only comparisons you can try to make are to things you understand. Right. And then you all you can come away with was, well, it's not that.
Right.
And we do this in more recent events too. I'm thinking of the discovery of prions.
That's right.
Yeah. It's not this, it's not that, and then we finally come around to what it is.
Well, yes.
But even
I still have friends who are keeping their fingers crossed behind their backs.
I mean, even what it is, a few years ago, I made a video saying what are viruses living, and I said, first, what are the characteristics of life? And I listed 5 characteristics, and Carl Zimmer got mad. He said, you can't define something by what it is. You have to have a definition.
Yeah.
Right. So even though you may have what viruses are, it's still not enough. You have
I think the whole debate about what is living is one that's gone on since Aristotle, at least. Yeah. And because it's, it partly has to do with what we think is important. I mean, Aristotle made the whole thing about what are essential qualities and what are secondary qualities. Mm-hmm. And philosophers debate whether we can make that distinction.
Philosophy was science at one point, right?
Yes.
Well, I mean, I like the definition. I know it when I see it, right?
Yes, right. Well, that's sort of what Andrei Lvov wrote this famous thing called "What is a Virus" in 1950, and he just said a virus is a virus. It's its own kind thing. And I mean, now if you ask someone what a virus is, they'll give you a whole series of attributes,
right,
and a whole series of sort of experiments. I mean, this is one of the things that struck me.
Depends on who you ask, though. A lot of people, you ask them what a virus is, they say, "Oh, you take an antibiotic to get rid of it."
That's right.
Speaking to scientists,
what would your definition be of a virus?
Well, I think my definition would be partly physical and chemical and partly functional. But I was struck by. I spent a sabbatical in George Klein's lab trying to learn cellular immunology at a time before in Stockholm, and at that time there were these things like killer T cells and so on, and they were defined by the fact that they had cells that came out of an animal. They were put through a column of nylon wool. They stuck to the column of nylon wool,
right,
or they weren't, and then they panned in a plastic dish, so they stuck or didn't stick to plastic of a certain sort, and then they bound to some, phytohemagglutinin kind of thing, or they didn't. And when they all came out at the bottom, these were cells that, when you put them back into the animal, did something. But it wasn't a very satisfying way to describe them. I said to George, well, what are the molecules on the surface all about? How do these cells differ? And he says, we aren't ready for chemists for another 20 years. He was wrong on the thing. It was about 3 years, but
today we define them by facts.
Right.
cell surface molecules, but I think it's still unsatisfying.
Right. Well, I mean, they were ones that protected the animal against tumors, non-specifically or something like that. Or they would lyse cells that had been sensitized. So they had an assay at the end, but they couldn't tell me what sticking to a nylon column really meant. It was they had survived certain selections, so to speak. And I think viruses were sort of that way. You know, they were things that had been passed through the filter, and they had been things that maybe were heated to 55 degrees and didn't die, and they survived antibody treatment of some sort. And so I think, you know, now we're down to, well, I think I remember, Rich, you wrote a piece or said something about the whole business of virus classification at one point.
Right.
And, you know, now we're stuck with a gazillion sequences. And what do the sequences tell us? We thought that maybe something like that would allow us to make sense out of this. But the more we look, the more it seems like, things trade genes around, and we can't really say that this is pathognomonic of anything, you know. But this is true in medicine. I mean, the 19th century was, the historians of medicine often view it as sort of the great time of nosology. That is, the great French clinician Pierre Louis famously said something like, "There are no diseases, there are only sick people." And you know, the idea is that tuberculosis is a thing now, whereas it used to be you would have a quartan fever or a tertian fever, which would be descriptive of how you were sick, but it has nothing to do with the etiology, if you will, of things. So the thing changed from a fever to a microbe. And now we even equate the microbe with the disease, the other way around. So.
I like this 1922 H.J. Muller, which you point out. He called phages naked genes. Well, of course, they're not naked, but I like this idea that they're just genes that move around, right?
Right.
And they have, if they're going to multiply, they have to get inside of a cell. So I like that.
No, that was a very famous quote, because a lot of people picked up on that. Because Muller was sort of an iconoclast, and I mean, he was a Drosophila geneticist by his profession, but he was a wide-ranging thinker. And
by that time, I mean, when phages are discovered, is it clear that they're viruses?
No,
not at all.
No, I mean, that's just Bourdais' argument was that they were somehow cellular products that were stimulated to be produced or induced, we'd say, by the outside phages. So that was his model. We now have an idea how he came to that, because he sent one of his associates to, there's letters about this, and again, why you should go to the archives and read this stuff. There are letters where he sent a student or a postdoc to Durrell's lab to get a strain, and Durrell gave him a strain the host strain, which turned out to be lysogenic. And so he was working on the lysogen. They didn't know what lysogenic strains meant at the time, but it turns out later they found that it was a lysogen, which is a kind of strain where it's got a virus hidden in it in a quiet form, and you can induce the virus to be produced. So it caused all sorts of messy experiments. And then there was John Northrop, who was the Nobel Prize winner for studying activation of enzymes from pepsin to pepsinogen to pepsin by proteolytic cleavage. These enzymes are inactive in one form, and they become active. They can self-cleave. And he and his colleague Albert Kruger, who was at Berkeley, they believed that phages were just an example of autocatalytic activation of something already in the cell. And so you put some phages in, and there was always already the cellular product that could be reactivated, which accounts for sort of the apparent growth of phages.
When would you say that we realized phages were viruses?
Well, I think we have it has a lot to do with when we developed ideas of what viruses are.
Of course.
So, I would say that somewhere between the 1920s and the 1940. I mean, the electron microscope really changed things, because people believe what they see. They see these particles attached to the E. coli cell, and they all have a standard morphology. You know, they're
particulate.
They're particulate. I mean, Virol actually said he talked to Einstein when they were both at Leiden in the 1920s about what a plaque assay meant. And of course,
so by then we had plaque assays.
Well, Virol
developed the first one.
The Plastax. Okay. That's what he noticed, these virgin spots on the lawn.
And he could dilute out and see.
And they diluted exactly according to dilution. And he talked to Einstein, and he said they did basically a dilute in tubes and find which tubes are infected, and it followed a Poisson distribution. And Einstein, of course, had worked on diffusion and all sorts of things like this. And Virol says in a footnote in his book that he's talked to this about to Einstein, and he said Einstein agreed with him, and Virol said something to the effect that, and he's a great mathematician, and it's very hard to satisfy a mathematician.
Well, you know, this sort of event, this time, as described in your review, the Inventing Viruses review, I found really interesting because if I've got this correctly, the fundamental conclusion from that sort of dilution and plaque assay experiment is that this agent is corpuscular in nature.
Right.
Okay.
Yeah.
And I love the word and the concept because they didn't know what it was.
Right.
All right. But corpuscular, I guess, it's not quite saying that it's particulate, but it's got that implication. And then it's not, what was this, around 1920 or something, or even before?
Right.
And it's not until 15 years later or so, in the mid-30s, that they can actually see these things in the electron microscope.
Well, I think the word corpuscle was more widely used colloquially, too. I mean, talk about red blood corpuscles. And white blood corpuscles. And it just means little body, corpus and ULE.
Okay.
So I think that you have to look at sort of the current jargon vocabulary that people used. And so
the corpuscle is the individual cell in
red blood cells.
A white corpuscle, a red corpuscle, and then these viruses are their particles, their corpuscles.
Yeah.
So when they see phages in the electron microscope, then they're particulate. How soon after do they see animal viruses and make the connection that?
Well, I think they took. We had Wendell Stanley, as you know, crystallizing poliovirus and TMV.
Right.
And I think right away, once they got an EM, the RCA, I think they took pictures of those crystals.
And they saw the particles.
And then but figuring out how to prepare the samples. I remember one of my teachers in Wisconsin, Paul Kaysberg, spent a lot of time figuring out how to shadow and stain, negative staining and so on. It's just, what you're seeing, of course, is not the particles themselves, unless you do cryo-EM. You're looking at a metal shadow of it, whether it's osmium sloshed around on the thing or whether it's metal sprayed, as bits. And
I don't know if you, when you were at ASV this summer, did you see there was a little exhibit of a centrifuge?
Yes.
Who was the guy who invented it? He was from Wisconsin, I think.
Well,
you remember that, Rich?
No, I didn't see that.
It was in the lobby at the
it was building. It was a multi-sample rotor. It was really
took a bunch of pictures of it. Bakke was his name, Bakke.
I think he was at
Indiana or something,
Michigan.
Yeah, and he developed this rate zonal centrifuge so you could purify viruses and so forth. So you note in I think in your talk in 1928 the first book is published by Thomas Rivers, kind of the father of virology, right?
Well, he was head of the virus research at Rockefeller. And he was professors had a lot more charisma in those days. I mean, there were leaders who everybody looked to, and they were patrician in many ways. They had enough money to spend their summers at Woods Hole and so on. Or even like the guy who founded JBC had his own private lab here in a penthouse in New York.
Yeah.
But he got a bunch of authors together to talk about what was known about filterable viruses.
So that
and the book was called Filterable Viruses.
Yeah.
So it was no longer a chapter in a microbiology book or a bacteriology book or something.
But there's still this idea that they're filterable. There's all viruses still, and these are the filterable kinds.
This is right. Because they wanted to basically focus on them rather than every other bacteria you could think of. Yeah.
So when did filterable get dropped?
Well, it seemed to get dropped about just right after that.
After that,
yeah.
Okay.
I mean, I think forget there was somebody. I think it was Bain Jones and Zinsser in one of their textbooks in the late 30s said, let's drop the term filterable because everybody just uses it as viruses, and we don't use virus for bacteria anymore. And so,
when did that stop? Do you know? Not using virus to describe a bacteria.
I think somewhere probably in the 19-teens, for sure.
Now you mentioned Stanley. This is important. So he crystallizes this tobacco mosaic virus, and he says it's all protein. There's the 5% phosphate he ignores, right? Which I think you mentioned at your talk. You just ignored it.
Yeah, well, he was a chemist,
right?
And he was an organic chemist, and I think he just felt that he knew that you get contaminants, and phosphate is pretty ubiquitous in buffers and so on. I remember talking to Bill Peary, the guy Bauden de Peary, who actually found that the phosphorus was in an RNA molecule.
Yeah,
and he just said, well, he didn't like Stanley very well. He said he was just a bad chemist.
I've heard that before, yeah. Well, he thought it was an infectious protein, right?
Yeah.
Because we're not thinking about nucleic acid at this point.
Right.
In time. This before people thought nucleic acids were important at all.
Right. Well, the nucleic acids were, if anything, were structural because they didn't, the Levine hypothesis had it as a repeating tetranucleotide.
So what year is this now?
Stanley?
Yeah.
36, I think.
Yeah,
36.
Yeah.
And when were Oswald Avery's experiments? That was like early
47.
40, in the mid 40s.
Mid 40s, yeah.
So we don't understand DNA as the genetic material until like 10 years after this.
Yeah, even then people didn't buy it, right?
Right. Well, there was Griffith's work in the Rockefeller in 28 on transforming principle. And that's all through there. You mean, you read the Mac McCarty's book on the transforming principle, which is an amazing book. And he said, they were sort of convinced that this nucleic acid component had something to do with it. But the problem was how to show it conclusively because they had contamination issues all the time. And when they finally got DNase and RNase as enzymes, they could show that, they could rip up every bit of DNA that was there. And that took it out. They put RNase in and it didn't touch it. And proteases, they finally had the tools, again, the technology to really drive the thing in.
Erling Norby was here last week. Do you know Erling?
Yeah.
He's written a bunch of books on Nobel Prizes. And he said even when DNA structure is solved in 1953, it takes many years for people to accept this, and they didn't get their prize till the 1960s.
Right.
So.
Well, I did a Google Ngram at one point on, the number of times the word appears and so on. And you look at what references were made to DNA. There was really no significant things you could do with DNA until hybridization came along. And that allowed you to, I think the picture of Jim Watson and pointing to the DNA molecule is instructive. That's all you could do with DNA is point to it. No, you could say this is explanatory. This is how the genetic copying mechanism could work, but we had no idea how to do it. And Paul Doty and Carl Schulkraut and Julie Marmer figured out how you could take the strands apart and put them back together again and test whether they would go back together again. And they had this amazing couple of papers about taking DNA from different sources and seeing how much they went back together. And they were doing bacterial taxonomy that way. And I think that was a very underappreciated piece of work. I wrote a paper about it, but I never could get it accepted. So
I think it's important to point out that viruses were important in this whole idea that DNA or RNA is genetic material. You had tobacco mosaic virus being taken apart and put together. The RNA is infectious. Yep. Frankel Conrad, right?
Yeah,
and
right.
Others.
Yeah.
And then, of course, Hershey Chase, showing that's actually the DNA that gets into cells and starts the infectious cycle. So that was a key part of it.
Mm-hmm, mm-hmm.
Right. Now, the other thing I wanted to ask you about was Delbruck. So he starts a phage course. He decides he's a physicist who starts to tell us about what he was trying to do.
Well, Delbruck was a physicist from Germany and son of an eminent historian, professor in Berlin. He's non-Jewish, and he worked for a while with Bohr and in England before the war on his physics PhD. His advisor was a woman named Lisa Meitner, who I think got the Nobel Prize, if I'm not mistaken. And he somehow didn't pass his PhD exam the first time around. And whether that contributed to him looking for other pastures, I don't know. But he was working on what people said was sort of minor problems of scattering things. But he was a smart guy. And I think, being a student of Bohr was a wide-ranging intellect. And I think that's an aside, I should say, is I think that we don't appreciate the fact that this differentiation of study of matter into what the physicists study is the hydrogen atom and the biologists study all sorts of complicated molecules. I think the physicists thought that anything having to do with matter was their province before the war. So you had these physicists working on proteins they didn't see a distinction between a protein and a hydrogen atom in terms of territory of field. So Bohr, I think he just ranged over all this. His father was a hematologist, basically found the Bohr effect in hemoglobin. And, but as I say, that's an aside. But I think that he got interested in what a mutation was. I mean, these mutations seem to be very bizarre. Why a mutation should be, and they were studying Drosophila, why they should be so stable and yet so infrequent. And how could a molecule or anything inside the cell be that stable? And because they were looking at molecular or atomic transitions, quantum jumps. And those kind of things were very stable but very rare. And so they thought that this might be the basis for mutation.
I see.
I mean, why a mutation is 10 to the 9th, frequency or something like that. And yet once it's there, it's there. And so he and a physicist, he was a physicist and a radiation biologist and a Drosophila geneticist in 1936 wrote this paper about sort of a quantum mechanical model of genes. It became a very famous paper, although not widely circulated. Delbruck wrote the sort of physical kind of interpretation of this. Because Muller had found that radiation caused mutation in Drosophila. So this was an explanation that they were working on. He then got interested in biology at this level, and he got a fellowship to go to Caltech, sponsored by to work with the Drosophila geneticists there. And when he got there, he found that Drosophila genetics was too complicated. I mean, I think those of us who tried to learn Drosophila genetics realized that, again, it's like the cellular immunology. It has a lot to do with, the history of a mutation.
Complicated.
I mean, bithorax. I mean, for God's sakes, how do you understand that? Or then having to learn all these mutations about wingless and this and that, and it was lore. And so he went there, and he was working with the Drosophila geneticists. And one day he heard that a guy named Emery Ellis, who's working in the basement on phages, had given a seminar, and he missed it because he was probably hiking in the desert like he was wont to do. But he went down to Ellis and says, "Well, tell me what your seminar was about. And he saw these plaques, and he says, "Well, these are the atoms of biology, because you're really measuring one over Avogadro's number of molecules." Mm-hmm. And Ellis had gotten in there for a whole other series of things. He was supposed to be doing cancer research, and they didn't have any money to work with animals or animal viruses, and so he decided phages were the way to go with a model system to learn how viruses replicated in general. And
so, what was Delbruck was at Caltech in what capacity?
Well, he was there as a postdoctoral fellow of the Rockefeller Foundation.
Okay, and what year are we talking about now?
1938. This is before the war.
That's even before I was born.
And he and Ellis published a couple of papers. Most people think that Ellis was the postdoc. It was the other way around. Ellis was working on this because during the Depression, he didn't have a job. And ever since he was a freshman at Caltech, he had been a sort of a favorite of Robert Millikan, the president of Caltech. And so after he got his PhD in physical chemistry, he needed a job. And Mrs. Seeley Mudd of the Mudd family wanted her son back in California after he'd gotten his training at Harvard Medical School. So she funded a cancer research division at Caltech for her son. And Emory got this job as his sidekick to do the lab work. And things were going well, and they were doing this kind of stuff. There was a lot of sort of side issues, but then Ellis, like many of the people at Caltech, got upset with Mudd because the Mudd family during the war, they had much of their money in mines in Crete and Cyprus, and they were cutting a deal with Hitler to leave the Mediterranean islands alone in exchange for the Mudd family being their advocates of Hitler in America. And the Caltech faculty basically threw them out. And so all of a sudden, Emory was without a job. He then, again, I think having the right people look out for you, he got a job with the Navy in ordnance, and he became the director of the China Lakes Project and became the inventor of the Sidewinder missile. And when Harold Brown was the Secretary of Defense, Ellis was brought to Washington as his head weapons analyst. So he had a whole nother life. People wonder what happened to him. Well, he had a whole nother life in a different field. I traced him down, and he was living in a house trailer up above Bakersfield, and spent a couple of days interviewing him on a couple of different occasions. And, he and his wife just chose to live there because they liked that sort of rural place.
Because I'm thinking of Ellis and Delbruck, famous one-step growth curve.
Yeah,
right?
Yeah, right. Well, the famous one-step growth curve was really just a repeating of a one-step growth curve in Durrell's work. Mm-hmm. And it was in a table, and I replotted the table, and it looked just like a Delbruck-Ellis one-step growth curve. So what
we're talking about is the growth of phages in bacteria. You infect at a high multiplicity, so all the cells are infected.
Right, and they go through a timed cycle, right?
Yep.
So again, it's like an embryological experiment, as I said. They develop from infected to mature phage in 10 minutes, 30 minutes, something like that. And then the whole cycle starts over if there's more bacteria.
So at what point does Delbruck decide that this is what he's going to do for the rest of his life, and why?
Well, I think he decided that this is the key to understanding reproduction. Okay. Because the phages reproduce in 30 minutes, and it's clean. It behaves like a black box. Physicists like black boxes, input, output kind of things. And but then his fellowship runs out, and he's supposed to go back to Germany, but by then the Nazis have taken over. And again, friends in high places, the Rockefeller Foundation manages to get him a job at Vanderbilt in the physics department, teaching introductory physics. But
just too far from Oak Ridge, by the way.
Yeah. Well, as it turns out, Luria, who was here at Columbia, had to go to Indiana because alien scientists were not allowed to be in the coasts. They had to go and be at universities in the middle. So the Midwest universities actually
that made a lot of sense.
Yeah. Well, they were afraid they'd
something
bring the subs in, and
I didn't know that.
Yeah. That's the Midwest universities really benefited from that kind of discrimination. And they got all these emigre scientists,
Yeah.
I mean, Chicago is where Fermi and all those guys were.
Right.
It wasn't Harvard or Hopkins or Cal.
Interesting.
Yeah. So he spent time there and said he spent most of the time collaborating with the microbiology department.
Mm-hmm.
But then Caltech offered him a position and he moved back to Caltech as professor.
So at some point he starts this phage course at Cold Spring Harbor.
Right.
What is he trying to do there?
Well, I think, again, he was a proselytizer, a discipline builder, and he felt that they really needed to convince other people that this was a good model system.
Mm-hmm.
And he also wanted to indoctrinate him the way he wanted him indoctrinated. I mean, it was very much part of you have to learn the rules of the club.
Yeah.
And, I'm working on a book on the history of the phage group right now.
Oh, cool.
And one of the chapters is really what constitutes a group, because they had this phage newsletter and called the Phage Information Service.
Right.
And all the copies are in the Caltech archives. And in the margins, you see Delbruck has written down who's going to be on the mailing list. And people come and go. People get dinked, and then people get added. And I remember once, I've asked several people. I asked Seymour Cohen, who was a biochemist, about, were you in the phage group? And he said, no. He said, I went to all their meetings. I was always there, but I wasn't in the group. and they kept talking him. They referred to him in private correspondence, very derogatory, like he's a biochemist.
Or they could have referred to him as a fruit fly on the wall.
Well, that's right. Well, he came out of the enzymology group of the Rockefeller. The other person was Lloyd Kozloff, who was from the Chicago group. Now, the Chicago group, again, were biochemists. And I once asked Lloyd, what made you in the phage group or not? And he says, you were in the phage group if you sent your preprints to Max for approval before you submitted.
Wow.
And so you have an operational definition of being, and as a matter of fact,
so much for the rationality of science.
Right. Well, come on. That's what historians make a business
this
at.
Is great.
Trust science, don't trust scientists.
Yeah.
But my colleague, Larry Holmes, wrote a book about the Mendelssohn-Stahl experiment. And the first chapter is about the double helix paper. The double helix paper went through, I think, over 8 revisions, all of which Max weighed in on before they submitted it, all in the course of 6 weeks. I mean, it was just back and forth about the language and
interesting.
The famous phrases of like, it has not escaped our notice. And, were they going to put it in that phrase or were they going to say it some way? I mean, it was all very crafted.
Oh, I didn't know that.
When did Delbruck and Luria join up? And what were the circumstances behind that?
Well, that, again, is a story is sort of interesting because Luria, as everyone knows, was from Italy, and he started to work on phage there. And being Jewish, he decided he had to get out, and so he went to Paris, and he worked with a very polymath physicist named Fernand Hollweg, who was in the Radium Institute. And he worked on the radiobiology, because he'd trained as a radiologist in Italy on the radiobiology of phages. And, but then when he quickly decided he had to come to New York, he came to New York and, he'd first written to Ellis, saying, because he'd read Ellis and Delbruck's paper and he thought Ellis was the person who ran the lab, which he was. And he said, do you have a job or a position for me? And Ellis says, well, I'm just sort of a research associate here. I don't have anything to offer besides which we're probably going to shut down. And he says, maybe you want to talk to Delbruck, And, so Luria had sort of gotten hooked up with Delbruck through the Luria thing. And, they met, I think in 1940 in New York here at some scientific meeting. And Luria was here, mainly because Fermi had written a letter for him to a guy named Francis at Columbia. He was a biophysicist. And so he was at Columbia here for a year or two, again, working with the biophysicists on radiation biology. Mm. People forget that radiation biology, before tracers, before you could do atom chasing, and labeling. It was sort of the way you found out what went inside the cell, because the physicists found you could go what went on inside the atom by throwing stuff at it, scattering. And they said, well, you can find out the nature of chromosomes or enzymes or whatever by scattering, or by what's the inactivation cross-section, if you will. So there was an awful lot of radiation biology going on.
So there's a biochemist here by the name of Shemin who became famous.
David Shemin.
Yeah, that's right.
Yeah.
I took a course with him, actually. Okay. I didn't do well either.
Yeah.
But that's another story.
Yeah.
But he got famous for just using a radio tracer to see how the heme group was made, right?
Right. No, so right after the war, well, actually, before the war, they had Columbia was quite lucky. They had some of these isotopes, and some of them actually were stable.
From Brookhaven.
Yeah, they were stable heavy atom isotopes, which you could measure by, specific gravity of a protein. They were very hard to get. I mean, not anybody could get these things. They had to be made for you, and to put them into molecules that you wanted them in was really hard. Yeah. And a colleague of mine at Princeton has written a book about the post-war traffic in radioisotopes, that how Oak Ridge especially decided that they would become the center of giving out isotopes for academic research. And, they had big greenhouses where they'd grow things in a CO2 atmosphere and things like that.
So I realize I just came in the middle of all this. This is non-virological, but the establishment of probably the epicenter for ecological investigation, science ecological investigation was at the University of Georgia, and they located there because of the Savannah River Project.
Okay, yeah.
Because they had access to all the isotopes in nature.
Well, yeah, it was really.
Odom was the guy who did that.
Yeah, it's okay. Yeah, I remember the name. But again, I think technology has a lot to do with.
So luck favors the prepared mind.
Right.
So you also told this interesting story. You call it Delbruck's subversive animal virology, where he gets a grant from Caltech.
Oh,
tell us that story.
They were always, like now, scrambling for money. Totally different scale, but you had to get money from the outside because the schools weren't paying, and it turns out that people always want to give money for diseases that affect them and so on.
Yeah.
And Caltech got some money from somebody who was interested in animal diseases, or human diseases. Now, Caltech, although they had this cancer research unit with mud for a while, that didn't take. But somebody came to them and said, we want you to work on animal virus diseases. And apparently Delbruck had two people. One was Delbecco, and the other was forget who.
Benzer.
Oh yeah, Seymour Benzer. Now Seymour Benzer was a physicist, and hardcore physicist, whereas Delbecco was a physician. And I think Delbecco saw more, had more affinity to this. And he had been working on phages. I mean, he'd worked on multiplicity reactivation and, again, ultraviolet biology, photoreactivation, I think he had a paper on. So he said, well, I'll go and see what we can do. And so one of the first things they did after he sort of surveyed the field to what it needed was they said, well, we need a plaque assay. And so fortunately, John Enders had just figured out how to grow tissue culture well, and do some plaque assays, or they didn't do plaque assays, but they were growing virus in tissue cultures instead of in animals. And so Delbecco then switched into animal virus work.
Right.
And it was sort of, I think, well, we have this money, what are we going to do with it kind of thing. And
I just didn't, I hadn't realized, I knew Delbecco had developed the plaque assay for animal viruses, but I didn't realize that Delbruck recruited him to go out and say, what do we need to do here? And you said that Benzer said, no, I don't want to do that.
That's right. That seems to be what their stories that they tell. And he had this meeting, again, he was a guy who built, communities, and he had this meeting called Viruses 1950, I believe, where he tried to get the people in plant viruses and animal viruses and phages together to talk about what the state of the field was. Mm-hmm. It didn't seem very successful.
Didn't work, yeah.
But he was always, he was big on taking his group hiking in Joshua Tree to build community. And he once told me, which turned out not to be true, but he once told me that he tried to schedule a phage meetings at the same time as the Genetic Society of America met. So you'd have to choose your loyalties. So you couldn't dabble in Drosophila genetics or be a Drosophila genetics and dabble in phage. Now, I checked to see whether that happened. It wasn't very systematic. A couple times it happened.
Dolchansky wasn't much of a hiker anyway.
Right.
One of the important points also you make is how many virologists got trained in phage as a consequence of all this, right?
Yeah.
And that's in part because of the phage course, trained a lot of phage people.
Well, I think the phage course was the sort of thing that people thought, oh, they should do this. They should find out what's going on. So you look at people who were professors or in another field. Richard Roberts took the phage course. I think he was a physicist, and there were people who didn't go on in phage, but they came, I think, just because of Delbruck's reputation and again, that it was a sort of new thing. And in 2 weeks, 3 weeks, you could get brought up to date and have a few experiments. And the interesting thing is, if you look at the people to the first phage course, there were, I think, 8 people. There were 2 women and 6 men, and all the men were associate or full professors. One woman was a research associate at Michigan, Harriet, forget her name, last name. The other was a woman named Phyllis Margretton, who was had just gotten her BA and was a lab tech at at the University of Illinois. And I was wondering why was she there? Because, and I traced her down, and she's just died. And her son is a professor of physics at Cornell. And it turns out her grandson is a professor of biophysics at Yale. Right down the hall from me. I didn't know that because the names are different. But anyhow, they said, yeah, she got this job as a lab tech and in a chemistry department. And the chemist professor was interested in finding out what was going on. So it sent her to this thing. She ended up getting a PhD in something else and had another career. But, I think this was a kind of proselytizing thing and also advertising.
So the phage course on your slide here. So this phage course was taught at Cold Spring Harbor during the summer, right?
Yes.
Mm-hmm. You have between 1945 and 1970.
Yep.
Wow. And was it always Delbruck and Luria who ran that?
No, Luria didn't have much to do with it except give occasional lectures. Delbruck ran it for a while, and then he turned it over to his, people who had trained, so to speak. He got interested in the late '60s in neurobiology, and gave up phage. And he, about the time I first met him, he told me that if he had, I think the Watson-Crick paper sort of did it for him. He said, if I had known it was going to turn out to be as dull and boring, I wouldn't have worked on it. Because it's just stereochemistry. It's base pairs fitting together like pieces of a jigsaw puzzle. He was looking for something big and important, not just stereochemistry. And I think that's overstated.
Delbruck was responsible, was he not, for popularizing this assembly of the T phages? Didn't those come from him?
Well, right. So the feeling was that they needed a set of phages that everybody worked on, because up to that point, one group worked on one phage that they'd gotten from their sewer and another from somewhere else, and they'd get different results. And like any of these things, you always ascribe your aberrant results to strain differences.
Right.
And I think he said, well, we got to work on the same set of phages. And they picked actually, the source of the phages were sort of interesting. A number of them, 2 or 3 of them were phages that had already been discovered. And Al Hershey had gotten one of the old phages, which became T2, from the guy he worked with long before that. But they went to a lab in Long Island one day, and they got phages that were being grown for therapy at that point, and they were making phage cocktails.
Sure.
And Max told me, because I was particularly interested in T7, Max told me that they went there, and the guy said, well, we have this cocktail, we grow the phages up, all these many, many phages for different diseases, and we put them into a pot, and that's what we sell you. And he says, then the doctor went out of the room, and the technician says, well, yeah, we used to do it that way, but we don't do it that way anymore. We mixed them together and we put them all together and grew them up, and that's what we give you now. Well, of course, the fastest growing one took over, and they only had one phage in that whole thing.
Oh, Jesus.
And that was T7, which Delbruck initially called Delta, modestly enough. But
Delbruck, Delta.
Yeah.
So wasn't someone at Rockefeller responsible for phage typing also?
Phage typing sort of grew out of.
Is that Rebecca Lansfield?
What?
No.
I'm
thinking of something completely different. I'm sorry.
Thinking of typing streptococci.
Yeah, that's right.
Phage typing actually was done, popularized by a guy named James Craigie in Canada. But interestingly, the phage typing groups never seemed to overlap much with the phage biologists. I think they saw phages as a tool and not as a thing to study of itself. I've looked Sorry.
Was there some point in time where Delbruck or somebody decided, okay, we're going to call this T1 and this T2 and this T3, et cetera?
Right. There was a paper that published out of Cold Spring Harbor, which put the set of phages that they had gotten together for the course and stuff, and called them T for type. And they decided to make these 7. And that paper was published by Demeritz, who was the director of Cold Spring Harbor, and a guy named Ugo Fano, who was an interesting character. Fano was a physicist who had been a schoolmate of Luria's, and he was also an emigre. And again, Cold Spring Harbor had a big radiation biology group that had been going since the 1920s. And he was a physicist interested in radiation, and he was assigned there, I don't know, just as by some group in Washington that had to do with emigre physicists. And Fano was there for a couple of years, being one of their radiation biophysicists. And then he left and went to Washington for a while, and then he became professor at Chicago and had a career really as a theoretical physicist. But this one paper is really on the T phages is his claim to fame in biology.
And when was that?
This was 1943 or 44, I think.
And how did they distinguish one T phage from another?
Well, they were all phages that affected E. coli B, the same strain of E. coli, because they didn't want different host strains, and they looked for plaque morphology. That some gave big plaques, some small plaques. Then one of the first papers that they published was they looked for immunological cross-reactivity, and they could say, oh well, these T2, 4, and E6, the ones the T even phages just happened by luck that they all were immunologically related. Yeah. But also they found that you could find strains that were resistant to T1, for example, that were sensitive to the other T phages. Okay. And sometimes there were cross resistance to, we now know because of surface receptors, but for example, T3 and T7 seem to have common cross resistance and things like that.
So now all these Ts are double stranded?
They're all double stranded.
Are they tailed? They're all tailed, or?
Well, yeah, they're all. Some of them have, like T7 and T3, have little tiny
short tails. Yeah,
Potovirus.
Yeah, and others are longer.
The others longer. Yeah.
Okay.
Yeah. So, but they all made clear plaques because they were not going to use turbid plaques, and lysogeny was something that muddied the waters, literally. Huh. Yeah.
Alan's nickname is turbid plaque.
Oh, okay.
Wait, it's his blog. Okay. You had to speak into the mic. You really need.
You have to speak into the mic.
Yeah, please.
Oh.
I can't hear you.
I maintain a blog called the Turbid Plaque.
Okay.
So the bottom line here is that phage made a huge contribution to virology in general, and in fact, you pointed out that all 4 major virology textbooks of the mid 20th century were written by phage virologists.
Interesting. Yeah.
But nowadays, very few virologists are phage virologists, although it's kind of having a renaissance because of phage therapy.
Yeah, there was a phage meeting that Rye Young from Texas had in Florida about 2002 or something like this. And there were several people from NSF there trying to get an idea of whether there should be more money put into phage. And it was really because phages were becoming so important to the biotechnology industry, phage display libraries and vectors and so on, but they didn't have any people trained to know how to work with phage. Yeah. And they thought the funding for phage had gone down, I think, to 2% of what it had been in terms of the overall budget and so on. And so there was a need for people who could grow lambda, for example, and to high titers and or know what lambda was all about. And there was sort of a real worry that this was going to be, information lost. So,
in fact, there's a interesting phage guy at Yale, Paul Taylor.
Right? Paul Turner.
Turner.
Yeah.
Right.
Yeah.
What phage is on the wall?
Yeah. What is that one?
Well, I don't know. It's it looks like
it's a long tail.
It has a long neck and it's a icosahedral head with tails. That's an artist's rendition.
Oh, I thought it was a scanning EM that colorized.
No, it's not.
Okay, all right.
I have one more thing I want to chat about. There's a quote in your review paper. This is the archive annual reviews of virology. You write that it's interesting to note that the discussion during this period, this is the mid 30s, 40s, was almost invariably framed in terms of reproduction, locating the problem in the realm of biology, not replication, a term later favored by chemists and now part of the modern discourse in virology. So I was wondering if you could about that.
I think it's important to I think figure out what the words meant to the people at the time, and not take our understanding of the word and place it back on the people who were using them if we're trying to understand what they were doing. And I think the notion of replication didn't seem to be. They were talking about biology. They were talking about how like begets like. That was the problem. How do you get a perfect copy? And they were thinking about rabbits, I think, and
reproducing rabbits.
And they weren't thinking about stamping machines,
which that would be replication.
That would be replication. They, there was this notion about how proteins are made, about the template hypothesis, and things like this. But I don't think people
Alfred Mirsky.
People were not getting down into the molecular details at that point. And I think there was this divorce between the biologists. And a lot of these people had come from medicine and physics and didn't think chemically. I mean, Delbruck basically said organic chemistry was totally a mystery to him. And
ironically,
Crick said,
Shargoff was a chemist and blew the whole thing.
Well,
we were talking about him before.
Yeah.
So this is interesting that today most virologists say replication, right? Whether they're referring to DNA replication or virus replication, right? But after you talked about this at ASV, and I came back here and I had a copy of General Virology, which is the first edition of the Luria book, 1953, and you cannot find you cannot find replication, but twice in that book. But reproduction you find all the time, as you're saying, because they're thinking about rabbits. And he has this one sentence which I think shows the growing difference in the usage. And he writes, "The results suggest a special mode of phage reproduction in which the chromosomes are replicated by a zipper-like mechanism." So those are the two uses of the word. That's the way I see it. Today it's not used that way.
Well, I'm glad that this is being outed, because the sort of ambiguous use of the word replication has always sort of bothered me, but I didn't know what to do about it. So this is good.
Well, some people
now I have another word I can use.
Some people say reproduction implies sexual reproduction, right? But I don't think that's necessary.
No, not at all.
Right?
Not at all.
No,
certainly not.
You can reproduce a dollar bill, right?
You can reproduce asexually and sexually.
Yes, that's true. Yeah. So I think that's the way it should be. The two terms.
Right.
The thing I think about replication is you're making identical images of something. So you have a stamp, like a rubber stamp, and you're just
or you're trying to make them identical.
Yeah.
Well, no, but when you think of reproduction, you have 5 children in a family, and they all look different.
Well, one of the problems is that during the reproduction cycle of a virus, there's always replication of a nucleic acid. Okay. And so
there you go.
You kind of need terms to distinguish between those two.
I think so. In fact, I told Bill earlier, a few editions ago in our textbook, we decided to use distinct the two distinct words, replication versus reproduction. And we do that, and we're very careful about it. But
on the other hand, at least at our university, we have a reprographics department. We talk about reproduction of a photograph in some way, meaning some sort of copying, mechanism. But copying implies a kind of a mechanism, whereas reproduction is much more, agnostic about the mechanism.
Got it.
Also, if you had a department of reproduction, people would get confused.
They would. So,
well, I remember. Yeah. Well, that's an aside. But when we first had a department of gender at our university, people thought it had to do with the particular nouns of German language. But
so you said you're writing a book on the phage school.
The phage group, yeah.
Phage group.
Yeah.
Will that be your first kind of like history book, or have you written some?
Oh, no. I wrote a biography of Felix Virol.
That's right. I have it, actually.
Yeah.
Yes. What's the name of that?
Called, you have it, Vincent.
Felix Virol and the Origins of Molecular Biology, which was a dumb title, but that's what they wanted. I finished and edited a book about Seymour Benzer and his reconception of the gene that my colleague started, and I finished for him when he died. And I wrote a book about plague in Manchuria and geopolitics of disease, which is, again, historical. So, those are
okay.
This phage group is really, I think, taking the Virol story. What happened when his generation went out of the picture, and the generation that I knew came up, and not the generation, of course, that's around now, but
right.
So it's going to come up to about 1960. But this generation from 1940 to 1960 is, I think, when phages were having their heyday.
I was born in 1940. I was just a phage in the mind of my parents, perhaps.
When's the book going to come out? Do you know?
Oh, that's another issue.
Okay.
No, I mean, it's taken me about 10 years.
Always good to ask an author when their book's going to be done.
Yeah. My problem is that my parking lot is the same parking lot used by my editor at Yale Press. Oops. And so I get this kind of question all the time, when we pass in the parking lot. But there's advantages and disadvantages to being so close to your publisher.
No, I'm just asked because I'm looking forward to reading it.
Yeah.
I want to be out after it's done.
Yeah,
well,
right.
the problem that comes out is in a book like this, you begin to see more and more facets that you want to talk about. So I was almost done, and then I realized the question comes, why is Cold Spring Harbor so important? I mean, every scientific discipline that I talk to, and even non-scientific disciplines, people have a sense of where their intellectual home is in terms of space, whether it's Woods Hole for the physiologists or Cold Spring Harbor for the phage people, or, Pacific Grove for some of the
Scripps.
The medievalists, for example, have Kalamazoo. And
really?
How? Yeah, right?
Not the cloisters.
No. It turns out you ask why, and it turns out that's where Upjohn Company is, and the Upjohn family had a number of kids who went into medieval studies, and they funded a huge big conference there 50, 60 years ago, and it's replicated every year. And people say, "Oh, are you going to Kalamazoo?"
By the way, I'm sorry I missed the first half of this, and I want to publicly apologize for being late. Did you mention anything about Morris Pollard's perspectives in virology by any chance?
No, I didn't.
Would you ever?
I think
did it contribute anything to the?
Well, I don't think I know that book. I know of Morris. He was interested in germ-free animals.
He was, and I knew him because I was a student out there. But I know that every year he had this remarkable New York City-based symposium that lasted a week, and they had the Metropolitan Opera come in and all kinds of fancy dinners.
Well, I'll have to look into that.
And they were funded by the birdseed company,
Hertz.
Oh, Hertz.
Yeah.
That's right.
Hertz. Sorry, Hertz.
Like the mountains, yeah.
Not Hertz. Yeah, that's right. And all their wild birdseed was collected right over here in the Meadowlands in New Jersey.
Are you serious?
Yeah, dead serious. So that's what funded an enormously extravagant, and I say lush, party type atmosphere for a week with high-end virologists, really top-end virologists. So
I'll have to look into that.
I think you should, actually.
Yeah, high-end virologists, like a Porsche or something?
Something like that.
I know this meeting because when I was a student with Peter Palese, he went to one.
Yeah.
And he was gushing about it when he came back.
Right,
he said the birdseed people fund this, and a book used to come out of it
because Mars Pollard worked on psittacosis.
Oh, okay.
And that's how the connection was made.
Okay.
They collected the birdseed and the metal in. So, interesting.
Yeah, they got it for free, basically.
Okay, last thing. Let's do some picks. Bill, we pick things at the end here that we like. If you want to contribute, you could do that, or you can pick one of your own books actually if you want. But we start with Alan.
Okay.
What do you have for us?
So my pick, I know Rich will appreciate this. I think others will too once I explain it. This is every now and then I get these news stories about somebody raised some old vessel and they're trying to restore it and raise money to do that. And it's like, it's just some random schooner or whatever most of the time. But this is a ship that was raised off the bottom out in the Pacific Northwest. And they're trying to restore it, and it is best known for when it was a fishing boat. It was a purse seiner off the coast there. It was originally known as the Western Flyer, and it is best known for a 6-week charter that it did with a couple of guys named Ed Ricketts and John Steinbeck.
Them.
Which produced a book called The Log from the Sea of Cortez. And it is which is one of the great works of popular science writing. It's a wonderful, wonderful book that tells this story that captures a snippet of time in the of biology at that time. And Ed Ricketts was one of the pioneers of marine ecology. And it just they went and they took biological samples all around the Sea of Cortez and produce the book. So read the book, but also read this article, which is a really neat synopsis of what this ship has been up to and what they're going to do when they restore it, which is also very cool.
Yeah.
So I looked up Log from the Sea of Cortez was my TWiV pick, my science pick in TWiV 163.
Wow.
Also back in 2011.
That's pretty cool.
So what did the flyer, what was it doing on the bottom in the Pacific Northwest?
Well, okay. So as a scuba diver, I've come to the conclusion that the natural position for a fishing vessel is upright on the bottom. They all end up that way at some point, it seems. So this one, sure enough, it had sunk and was sitting there for a while, and somebody finally raised it, knowing what it was, and then tried to. I don't know what they were trying to do with originally, but then they sold it to this organization that's going to turn it into a research and education ship that will do cruises with students to get them in marine biology. Very cool. So it ended up on the bottom because it's a fishing vessel and they're in a high risk industry. Right. But
so is Ricketts related to the other Ricketts at the University of Chicago?
I don't know. I kind of doubt it.
It's an unusual name. I mean, they named the disease after him.
Right. No. Ed Ricketts was he wasn't an academic biologist. He was a sample collector who, which was, a kind of Wallace's line of work too.
Right.
So he collected samples for museums and also had some interesting ideas in marine ecology, which have now become standard, but which were somewhat revolutionary when The Log from the Sea of Cortez came out.
Mm-hmm.
So I was talking about Nobel Prizes with Erling Norby recently, and I was looking at a picture of the Watson-Crick ceremony, and that was 1966, I think.
Something like it.
There's a picture of all the laureates that year, and there's John Steinbeck.
Yes.
And I said, wow, would I have liked to be there?
For the Grapes of Wrath.
I bet they didn't even say hello to him, Watson and Crick. They probably didn't know who he was.
They didn't even know who he was. That's right.
How cool would that be to be up there with
that's amazing. Amazing.
Well, thinking of strange things about who knew who was working with what was that Watson was a student of Luria's at Indiana in the biology department. Also in the biology department at that time was Alfred Kinsey. And here, we wonder whether these two
that's
people
reproduction.
Two people at the different ends of the hall, whether they knew each other, were both sort of doing earth-shaking kind of work.
They sure were.
But maybe totally, ignorant of each other.
Of course, your colleague was a student of Watson, Joan Stites, right?
Yes, Joan. Yeah.
Yep. And Crick.
All right.
And Rich, you work with Joan, yes?
What?
Am I not right in assuming that Rich Condit worked with Joan Stites?
Yeah.
He did.
So, okay, there's a good connection here, though.
So when you were at Yale, Rich, you must have known Bill, right?
Yeah. I was getting to that.
Okay.
Did I, by any chance, serve on your
no,
dissertation committee or something?
You weren't on my committee. It was Dave Ward and Paul Howard Flanders.
Oh, okay.
And Joan.
Yeah. Well,
I don't know why you weren't on my committee, because in my mind you were basically my informal mentor.
Yeah.
Okay.
Well, our Joan's lab and my lab and John Cronin's lab, who's now in Indiana, Illinois, we all had a joint research conference once a week.
That's right.
And
that's right.
So our groups are really integrated.
The interesting thing, I mean, I did my thesis on T7. Joan was interested in developing that for her own purposes, but you were the source of T7, the phage, and also all of the technology and the lore. So, I mean, we and our other TWiV buddy, Ed Niles, was a postdoc in Bill's lab. And Ed and I became very fast friends. Okay, and it's always remarkable to me throughout my career. I've been on a pretty short leash, tied to my bench. But every day I would make this walk down the corridor and up a rickety old elevator. Radiobiology was what, 4 floors up?
Yeah, we were up. We were up in what used to be the intern's dorm.
Right. It was a great space. I loved that space. And and I'd visit Niles every day. He'd visit us every day, and it was just part of my life. So yeah,
I think the community of scientists was much smaller, and people knew each other more. I mean, you mentioned Peter Palese. Well, Peter and I and my wife and Mady, took a tour to China together and taught together there. And there was a time when, hardly anybody in molecular biology didn't say, oh yeah, I knew them from graduate school or we knew each other from Gordon conferences or something. Yeah. Now it's just so much bigger that
it is much bigger.
I don't think there's these kind of communities.
Sure. So Rich, what do you have for a pick?
So in honor of
that was all a digression from Western Flyer. As I was listening to that, I was looking at, and I was, wait, did I miss a transition? Are we now talking about Rich's pick?
No, we didn't miss it.
Not yet.
So in honor of Bill's visit, I decided to pick one of his papers from that time. And in fact, so we've talked a lot about T7 here. And the thing that I always think about is that probably anybody who's done any significant bench science, wet bench science in molecular biology has used T7 RNA polymerase or the T7 transcription system in some fashion or another, either as an enzyme or an expression system or whatever. And probably only a very small fraction of those people know what T7 is or where all this came from. Okay. And Bill was there right in the thick of it while it was all going on, contributing to the understanding of how T7 the T7 transcriptional program and ultimately the purification of the RNA polymerase that we, so many people use now throughout molecular biology. And there were a number of papers that I could have chosen. And if you look over the sort of the PubMed selection of papers, you'll see this delightful sort of progression of understanding of the transcriptional program in the phage. But a culmination of that is this paper that I've chosen, which is a paper in biochemistry by Ed Niles, Steve Conlon, and William Summers from 1974 called Purification and Physical Characterization of T7 RNA Polymerase from T7 Infected Escherichia coli B. So it's a great biochemistry paper that describes the purification of the enzyme and its properties, including how it transcribes T7 DNA to yield transcripts and et cetera. And, it's historic in so many ways. So I thought I wanted to acknowledge Bill's contribution to all of that history and technology through choosing that paper. And as I looked over it this morning, and it's got so many memories for me. But then I ran across this thing in the acknowledgments, and it says, the authors wish to thank Elizabeth Condit for a gift of hydroxyapatite.
Oh.
So hydroxyapatite was used as one of the purification steps. And Elizabeth Condit is my wife, who at the time was working in a lab, a dermatology laboratory as a technician. And for their experiments, she made hydroxyapatite, which is calcium phosphate. You could buy it commercially, but she made it. And Ed insisted that she made the best hydroxyapatite in the known universe. And the stuff that he could buy commercially didn't work nearly as well. I remember actually going over and getting hydroxyapatite from Ib to carry over to Ed. I showed her this morning, and she said, "What? I don't remember that."
That's great.
Well, it's there in black and white. It must be true.
Yeah. Great.
All I remember about that paper is Ed and Steve Conlon spending 2 years in the cold room because the enzyme was so unstable most of the time. And now when I go to the stockroom and see a reagent on the shelf, dried or something like this, I just said, well, that's progress, So,
what do you think? Now people buy this enzyme.
Yeah,
huh?
yeah. Well,
you probably never thought of it when you did this paper.
No, I mean, but there was a time when, again, technology important. If you were at Stanford and had Arthur Kornberg down the hall from you could do experiments that the rest of us could not do.
Yeah.
Because any old enzyme you wanted, you just go and now, of course, the tech companies make it possible so that it's more egalitarian, I guess. But yeah,
Dickson, what do you have?
Well, I had something I've been hanging on to for a while. It's NASA's recent declaration as to what we finally think our galaxy looks like. And it doesn't look as though it was the original long-arm spirals that we thought of. It's a barred spiral galaxy rather than a typical what you'd see out in outer space. So I just thought that as long as we're discovering how small and how interesting things look at the molecular level, we should probably take a larger view.
Yeah, it's a little curved though at each end.
It's a little curved at both ends. That's right. And
but it's not flat.
It's not flat.
And neither is the Earth.
Well, that's another discussion.
There's a guy who sings in my chorus who wears of course, in a chorus context, it's a pun, but he wears a T-shirt with a NASA emblem on it that says, "Not flat." We checked.
That's good. I like that. All right.
Yeah.
All right. My pick is a book by Rachel Maddow. It's called Blowout.
Oh, yes.
You read it?
I haven't. I know. I follow.
I watch her regularly.
It's a really good book. It's I heard it discussed on actually in an interview with her. It's corrupted democracy, rogue state Russia, and the richest, most destructive industry on earth, which of course is oil and gas. Very interesting history, and how it's corrupted almost everything on earth. It's corrupted the U.S., Russia, every other country, and if you're interested in fracking, it goes through the history of how they kind of figured out how to get that to work, what it's done to Oklahoma City. Really good book. I don't know how she got the time to write that, but
a lot of them put their names on Gross-written books.
You think so?
A lot of them do.
Oh, that's sad.
I know. I agree.
All right. Do you have anything you want to pick?
Well, I mean, you caught me sort of flat-footed here, but
okay.
This morning I saw on my news feed a paper I want to read, and that is a report of finally a reasonably successful dengue virus vaccine.
Oh yeah.
Oh yeah, I saw that too. It's a new one. Yeah.
Since this is virology day, so
you bet.
Yeah.
Okay.
So yeah, we should ultimately discuss that.
Yeah.
Yes.
There are a couple of others in the pipeline too.
Yeah.
It's interesting that there's so many, but yeah, the one.
Well, it's a worldwide problem.
Yeah, and the one that exists is not perfect. So
right, yeah, I mean, also I'm doing research in Southeast Asia now and on geopolitics of disease, following up my plague book, and I have a vested interest in keeping safe from dengue.
Indeed. Yes, indeed.
Yes.
Go in the winter.
Yeah, winter on Singapore
when it gets down below
on
90.
the equator, right?
Yes.
That's it.
All right, that's TWiV 573, microbe.tv/TWiV for the show notes, questions, and comments. TWiV at microbe.tv. If you like what we do, consider supporting us. You can go to microbe.tv/contribute. We have ways where you could give us a small amount of money every month. Give us $1 or so. And that helps us to take care of our expenses. Our guest today from Yale University, Bill Summers. Thanks for joining us.
Well, thank you for having me.
It's very interesting. You have a lot more stories, I suspect, to tell.
Well, when you get old, as some of you know, that happens.
I resemble that remark.
Yeah.
Do you miss doing research and publishing papers and all that?
Yes and no. I mean, you feel an obligation to your colleagues to keep up on one hand and show that you're still have it. But on the other hand, it's sort of free thinking like, no, I don't really have to, prove anything at this point. But what I do miss about having retired is not having the ability to teach a new course. I see a book I'd like to bring into a course or something, and I hear that. So that's.
Do you have any heroes in virology that you would like to share with us? Just take 3 of the top virologists. Who do you think are the top 3 virologists in your opinion that you really admire as a scientist and as a human being?
Well, I sort of have always admired Al Hershey for being a person who marches to his own drummer in many ways, and being a person devoted to his science, and there's.
Did you ever meet Peyton Rouse?
I did not meet Rouse.
I did.
Yeah.
Briefly. It was very brief.
Yeah. And, it's...
Or Bill Shope, Sr.? I know Bill Shope Jr. was at Yale, but...
Oh, yeah. No, I knew Bob Shope. But,
I met Bob.
Yeah, Bob. But not very well. And
did you know Jordi Casals?
No, I didn't.
Oh, right.
Yeah, I mean, I got into animal virology after a lot of these guys had done their work, So I was in the phage group then, and I mean, I must say that I think Jim Watson in his better days was somebody who I admired a lot. I'm sort of sad to see him go the way he's gone, but
Al Hershey's a good pick.
Yeah.
Someone just sent me a picture, by the way. They used to have a portrait of Watson on the wall in Grace Auditorium. Took it down.
Oh.
Okay.
Yeah, well.
Yeah. Dickson Despommier, thelivingriver.org, parasiteswithoutborders.com. Thank you, Dickson.
You're welcome.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. And Bill, it was great to spend some time with you.
Yes, well, good to talk to you. Yeah.
Alan Dove's at turbidplaque.com on Twitter. He's Alan Dove. Thanks, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.